Thanks so much for the introduction, Avi, and thank you for having me. As Avi said, my name is Anna Kelly-Nawan. I'm a software developer at the Pei Wang Lab at Mount Sinai, and I'm here to talk today about the ProTrack Kinase Activity Portal. It's a tool that we developed to help people explore the results from kinase enrichment analysis that we performed on the CPTAC data, which stands for Clinical Proteomic Tumor Analysis Consortium. And let's just jump right in. Cool. So the ultimate goal of this tool is to make, and the ultimate goal of the consortium at large is to make CPTAC results available to the scientific community. Of course, that can mean a really broad range of things. So I'm going to talk specifically about this kinase enrichment analysis that we performed on CPTAC Pancancer data, and a little bit about the preliminary results that we have in this in-progress study of different kinases that we found differentiating tumor immune subtypes, and finally walk through a demo of the ProTrack Kinase Activity Portal where you can explore these results yourself. And this is going to be the structure of my talk also, where I'm going to talk about why we focus on kinase enrichment analysis, go over a little bit of the data that we performed this analysis on, show some of the results that we have for which kinases seem to differentiate tumor immune subtypes, and then finally walk through a demo of how you can explore these kinase score results yourself and visualize them. So first, let's talk about protein kinase functions and what a protein kinase is and why we're so interested in this. Essentially, a protein kinase is something that takes another protein and catalyzes the transfer of a phosphate group from ATP to that protein, resulting in a phosphorylated protein. This is a particularly important process because it's a major driver of cell state, or major determinant of cell state. Changes in activity, localization, and stability affected by kinases can lead to changed cell states, including, unfortunately, in some cases, a tumorous cell state. This is of particular interest to us because kinases are small molecules that can be targeted and inhibited, hopefully preventing this process or arresting it. To this end, or as part of this end, CPTAC has performed really large-scale mass spec experiments, including the production of multi-omic data, proteomic data, RNA data, as well as really broad, really deep phosphoproteomic profiling. This is cool because that means we have these phosphoproteomic profiles across multiple cancer types, but what we get then is sort of a snapshot in time of the phosphoproteomic state of the cell, and it's not necessarily that helpful it's not necessarily like the ideal indicator for kinase state. Part of that is because you have some protein kinases that can phosphorylate multiple substrates, or you have, for a given substrate, multiple protein kinases that can phosphorylate it. But we do want to understand the kinase activity state because it lets us explore the following questions. Which kinases are activated or suppressed in tumors as compared to normal adjacent tissues? And in that vein, the ultimate therapeutic translational question is, can these kinases be targeted by small molecules as effective therapeutics? Or for that matter, be more directed in terms of how kinase inhibitors are administered. Okay, so now we talked a little bit about protein kinase function and why we're interested in them. I want to talk about kinase enrichment analysis 3 and this first question of which kinases are predicted to be differentially activated when comparing tumors to normal tissues. Of course, we could always use a crystal ball to guess this and estimate which ones, but we do have some more sophisticated statistical methods. Namely, some examples of some are expression 2 kinases, inference of kinase activities from phosphoproteomics, kinase perturbation analysis, and kinase substrate enrichment analysis. For this study, we chose KIA3, or kinase enrichment analysis 3, developed by the Mayan lab here at Mount Sinai. In contrast with other methods, KIA3 leverages many sources of publicly available knowledge for estimating kinase overrepresentation, including kinase substrate interaction, protein-protein interaction, co-expression, and co-occurrence data libraries, totaling 11 kinase substrate set libraries. So how does it work? it takes as input a phosphorylated protein list or encoded as human gene symbols, and then returns a ranked list of kinases. A kinase's rank is putatively is higher if its putative substrates overlap more with the input gene set. And essentially, this ranked list of kinases gives you just a relative sense of their inferred activity. So if you have already a list of proteins that you're interested in, you can just go ahead and get a return list of kinases from the Mayan Labs web application that's listed right here. And that's really great. That's really helpful and nice and easy and intuitive. But the issue, and I also wanted to mention that they also have appeters, which are Jupyter notebooks that you can download. They take GMT files and return these really cool visualizations like t-SNE plots, UMaps, heat maps, as well as the top consensus kinases. So the issue that we have, though, for the CPTAC data is that we have a really broad, diverse set of cancers and a diverse set of cases. So for a given, in pursuit of personalized medicine, for a given case, how do we choose this phosphorylated protein list? And that's what I'm going to go into next, by the pipeline that was performed by Weiping Ma and John L. Evangelista of Mount Sinai. So for a given case, we have phosphoproteomic profiles. And for each phosphocyte in that profile, we calculate the tumor over normal phosphocyte log2 folds change by taking the log2 of the tumor abundance of that phosphocyte divided by the normal abundance of that phosphocyte. Once we have the tumor normal phosphocyte log2 fold changes for all of the observed phosphocytes, we can rank them. And from here, we're able to take the top 500 phosphocytes and essentially what we're calling the tumor up protein list. This acts as the input for KIA3 for generating tumor up kinase scores. This gives us a ranked list of kinases that are predicted to have a higher impact on phosphocytes upregulated in tumor as compared to the matched normal tissue. Oh, did that jitter? Does that look crazy on your screen also?
Yes.
Okay, cool. Okay. All right, so let's do, well, we also did the same thing basically for the bottom 500 phosphocytes, which are supposed to be kinases that are more active for phosphocytes that are downregulated in tumor as compared to normal tissue. Let's leave this slide and see if things kind of re-regulate a bit. Okay, cool. And so we did this for all 5 cohorts basically that were published at the time. And I'm going to talk a little bit about those cohorts that we performed this kinase enrichment analysis on, where we performed this pipeline. And so I'll talk a little bit about the cohorts, which is what we chose. We chose the ones that were publicly available and published at the time by CPTAC3, the number of phosphocytes that were available, that were detected across all of the samples. And I am putting the number of phosphocytes in red because I think it's actually a pretty impressive number of phosphocytes across all of these samples. And it seems just like it's like a very rich and very large data set. I also want to mention that the samples themselves would total 378 paired tumor over normal samples, tumor and normal adjacent tissue samples. So this samples that I'm showing are not all of the samples that are available from CPTAC, but the ones that have paired tumor and normal adjacent tissue because of the pipeline that I showed earlier. This is really cool, and it's like another added benefit of the CPTAC data because other studies such as TCGA don't necessarily have the benefit of the normal adjacent tissue. So the cohorts that we've included in this particular tool include clear cell renal cell carcinoma. Please note I include the PubMed ID here for the flagship resource paper, where if you wanted to find out more about that particular cohort, you can go ahead and take a look at that paper. With about 28,000 phosphocytes across 84 paired tumor normal samples, lung adenocarcinoma with almost 33,000 phosphocytes across 102 samples, uterine corpus endometrial carcinoma with nearly 31,000 phosphocytes detected over 30 samples, colon cancer with over 17,000 phosphocytes across 96 samples, and finally head and neck squamous cell carcinoma with over 31,600 phosphocyte across 66 samples. Again, this is the paired tumor normal samples, and these are the phosphocytes that were detected in at least, this is already pre-processed, in at least 50% of the samples that we're looking at here. So we created kinase scores for all 378 of these samples. We calculated them and made them available in the portal. And something that I wanted to show, just some of the things that we found by generating these kinase activity scores, is that they can separate different cancer types. So that's pretty cool. When the samples are projected onto a t-SNE plot based on their enrichment scores, we can see that the different cohorts are in general clustered together very distinctly. So correspondingly, I mean, if you think then like, okay, this histological diagnoses suggest also like a corresponding change in kinase activity. The kinase scores are first ordered according to cohort or tumor type on the kinase portal. What's even more, if we go even deeper, is that the kinase activity scores can actually separate not just the cancers, but also the different immune types. So for the clear cell renal cell carcinoma flagship paper, one thing that we proposed was the separation of this tumor into 4 different immune types based on the tumor microenvironment makeup and the protein signatures. And these immune types are CD8 plus inflamed, CD8 minus inflamed, metabolic immune desert, and VEGF immune desert. And so one line of questioning that we're following is whether there are kinases based on these kinase activity scores that can distinguish one immune type from the other immune type. And this is just this like heat map showing the different kinases that can kind of distinguish between these one tumor type as compared to the others of the CD8 minus inflamed, CD8 plus inflamed, metabolic immune desert, and VEGF immune desert. And then we wanted to go a little bit deeper and ask, are there any dark kinases in this list? That seems like we could find out some more information about dark kinases while we're at it. And this one became very interesting to us, the PRKCQ kinase, which is part of this CD8 plus inflamed upgroup. And this PRKCQ was particularly interesting because, as you can see here in this violin plot, when we take just the tumor over normal protein abundance of this gene, of this protein over across all of the immune subtypes, it doesn't seem to really differentiate between any of the immune subtypes. However, in any significant way, if we do it based on the kinase score, what we do see is that it seems to be particularly enriched or active in the CD8 plus inflamed group. And that's really encouraging for just as a sort of sanity check, because PRKCQ is something that's primarily expressed in T cells. But beyond that is also just really interesting in terms of being like a molecule or a kinase that we can learn more about. And I did link here to the PRKCQ webpage on the Dark Kinome knowledge base. And what is particularly interesting after we found this is that it turns out as recently as June 2020, there was an article that a study that says that PRKCQ inhibition enhances chemosensitivity of triple negative breast cancer. And so the next question is, can it be studied for this immune subtype of this kidney cancer as well? And this is the type of study that we can do with these kinase activity scores, which just provides so much more data. And it's like just some sort of much richer insight than necessarily just looking at the protein tumor over normal abundance does. Okay. And now I'm just talking about one kinase for one cancer type, but obviously we've generated a really wide range of scores for multiple cancer types. So I'm going to walk through this website that I built for people, to the larger scientific community, to take a look at these results. It's called google.com. Just kidding. That was made with other government money, but I did want to show that it's live. So it's at pancancer3.cptacdataview.org, and you can just visit it with any web browser. And I'm going to just show a little bit of the interface. Up here, we have a link to some data about CPTAG. And then if you already have genes or proteins of interest, there's data here about the Mayan Labs Kia. You can get to the Mayan Labs Kia 3 page, which accepts as input a list of gene symbols for your phosphorylated proteins. On the other hand, what we have on this page is actually you enter a new line separated list of kinases. So the default that we have here are just some kinases that are part of the MAPK-ERK pathway. And what's automatically generated is this heat map with some clinical annotations, including tumor, survival status, stage, overall survival in days, grade, as well as the tumor up kinase activity scores that I mentioned earlier, whose higher scores indicates that it's more active in phosphocytes that are upregulated in tumors compared to normal, and the tumor down kinase activity scores. And then finally, we've also added this block here of the global protein abundance folds change, which basically allows you to see if the tumor over normal folds change, like if there seems to be more protein, but more of this protein in the tumor as compared to normal, and if that corresponds to the change in kinase activity score. So in this case here, we can see that like EGFR seems to have a higher expression in this particular cancer type and a correspondingly maybe higher kinase activity score. Okay, so let's take a look at the dark kinase that I was talking about earlier, which is a lot of letters that I can't fully remember. Okay, cool. And I did want to show that if you have typos or some kinase, like for example, some made up kinase that's not available in the database, it will let you know. And let's also look at a really well-studied kinase, MAPK1. And we just click visualize, and that takes us to this customize and interact tab, which has most of our functionality in here. And I'm going to talk through this functionality, starting with this view selector. So we're in this thing called the all kinase view, which just has that heat map that I showed with the clinical annotations, a tumor up kinase activity score, down kinase activity score, and global protein abundance folds change, which can also be really helpful because then we can also see which cancer type doesn't seem to, where like a given protein wasn't necessarily detected. And then I also wanted to show this thing called the substrate view. What this does is it creates a list of the kinases that you inputted, and it'll generate a slightly different heat map here. So you still have your clinical annotations, scores, protein abundance fold change, but what we also have then is the substrate abundance fold change for the purported substrates as listed by the KIA2 database. And so you can sort of see MAPK1 is a very well-studied kinase, and so we have a really long list of substrates here. But then you can also just take a look at what the changes in that substrate protein abundance is as compared to the kinase activity score. Cool. Okay, so we'll go back to the all kinase view to just walk through some of the rest of these features, including this little box here, which is this sort of sorting box. And you click an element on the heat map to enable the sorting. So I'm going to click the PRKCQ up track, and it shows me the series that I clipped, the underlying sample, and the value, which is really helpful if you are like investigating a particular outlying value. But what this also does is it populates this sort by functionality, which allows you to sort the heat map by any of the given tracks in case you're looking for a particular pattern. So let's say we sort by PRKCQ up, and let's sort it by ascending. And as you can see here, the kinase activity scores are sorted within each cancer in ascending order. And this can be really helpful if you just want to like quickly eyeball and see if there's a pattern that you're looking for. You can also see the underlying values with tooltips here. And as you can see, and like we mentioned earlier, it looks like, oh, in CCRCC here, the global protein abundance folds change doesn't actually correspond with the with this increased kinase activity score. So that's kind of a nice like reiteration of what we saw in the violin plot. So it's showing all of this increasing within each tumor, but what if we wanted to see what this pattern looks like across multiple tumors? You simply click this toggle here to unlock the tumors, and then click ascending. And now we can see all of the normalized kinase activity scores in ascending order and how that distributes across the different tumors. And this can be really helpful because like for example, right now we see that CCRCC seems to be really like dominated by these higher kinase activity scores. But if we take that away, you can still see that head and neck squamous cell carcinoma, this pink one, also still is like more heavily weighted in this tumor up kinase activity score for this particular kinase. And so I wanted to talk a little bit about these blocks on the left side, which is essentially like an interactive legend slash like light cohort query. So you can just toggle on and off some clinical variables to cut to like cut down or be more directed in your query. So now let's say, for example, I wanted to go down the line and see, does another pattern hold if I take out the head and neck squamous cell carcinoma? In that case, it didn't really seem like there was one. And so let's say, for example, I'm particularly interested in just high-grade, high-stage tumors of patients who were reported dead. You can just toggle all of these things on and off. And this is just like a really helpful, like lightweight filtering or query mechanism, but it can be, you know, quite meaningful. And so I'm going to show how the sorting functionality in particular can be really powerful here as an example in the substrate view. So we can see here again, the tumor up, it's sorted by the tumor up kinase activity scores. It does seem to be particularly like heavy in the CCR/CC end of things. But we don't really know which substrate, it doesn't seem to show which substrate is like really driving these increased kinase activity scores. And this is like where it can be helpful to be able to sort your whole heat map. So I'm just going to click tumor and then lock it so that I can see what's happening within each tumor, and then click ascending again. And here I can see, based on the purported substrates for PRKCQ, which ones seem to be driving this kinase activity score down here. For CCRCC, it seems to be particularly this substrate as compared to this other one, which is what's been detected. Cool. And then last but not least, you can download this plot as a PNG. And then if you are more computationally inclined and wanted to get some of this data for like some sort of downstream computational analyses, you can do a custom download where you select which worksheets to include. So including, let's say I want the tumor up kinase activity score, the PRKCQ, substrate protein abundance tumor normal fold change, and the global protein abundance fold change. You just click those, and then you will be able to download in Excel with those particular worksheets. And so I just want to show real quick, if I can get Excel to open, these worksheets. Okay, so we have the tumor up kinase activity scores. It's sampled by kinase. And then you can also see the global protein abundance folds change that's sampled by the protein or by the kinase as well. And then finally the substrate abundance fold change, which is the sample by the purported substrate. Okay, and that's the end of my demo. And I just wanted to thank all of the people who worked on this, including the manuscript co-authors. It's currently under preparation. All of the members of the Mount Sinai Data Analysis Center, and finally, all of the members of CPTAC, the CPTAC Consortium. It's been a real pleasure working with them.